Here we will explore true facts about the Gummi bear. Gummi bears are common in parts of Southeast Asia, although it is a well-known fact that these Gummi bears are often trapped by the children of Northwest Asia for their delicate furs.
In the arms of the angel, fly away from here, from this star.
Gummis has been around since the medieval times, and colors has been around since the 1950s. But the real question is, how have these prehistoric creatures stayed relevant throughout the times? Why, it's in their DNA, of course. DNA and RNA are a nucleic acid. Polymers are made up of many small repeating molecular units. These units, when put together, are nucleotides. When nucleotides are put together, they become a polynucleotide. DNA is made up of three things. Firstly, a sugar molecule. Secondly, a phosphate. And thirdly, one of four nitrogen bases. DNA gets its name from the first part of the molecule, deoxyribose. The genetic coding of the Gummi bear is found in the four nitrogenous bases, adenine, thymine, cytosine, and guanine. In living organisms, such as the Gummi bear, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. Basically, it's like a ladder that has been warped by some kind of large ladder-hating twister machine. A double helix! No, not that helix, although they have a few ragers that I listen to while I do my prancercising. In a DNA molecule, the sugars and phosphates James bond together to form a sexy set of twins. Backbones, that is. Those sugar-phosphate bonds I talked about earlier run down each side of the helix, but chemically in opposite directions. If you were looking at them, one would always appear upside down from the other. One strand begins at the top with the phosphate connected to the sugar's fifth carbon, ending where the next phosphate would go, with a free end at the sugar's third carbon. This creates the pattern 5' and 3'. On the other strand, the opposite DNA strand begins at the top with a free end at the sugar's third carbon, and the phosphates connect to the sugar's fifth carbons all the way down. It ends at the bottom with another one of those phosphates that I had mentioned earlier. It is called the 3' to 5' direction. Are linked by the nitrogenous bases via relatively weak hydrogen bonds. But they can't be just any pair of nitrogenous bases. If one nucleotide has an adenine base, only thymine can be its partner in crime. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2 bonds. It's the order of these 4 nucleobases, or the base sequence, that allows a Gummi bear's DNA to create the 4-legged creature of cute. Gummi bear? Oh wait, no. Who wrote this? I am positive that is not the correct way to say that. Anyways, Gummi bear chromosome 1, the largest of all its chromosomes, contains a single molecule of DNA with 247 million base pairs, and each somatic cell has 46 DNA molecules tightly packed into its nucleus. Since each nitrogenous base has only one counterpart, you can use one base sequence to predict what its matching sequence will look like. So, could one make the same base sequence with a strand of that pesky RNA? Nah. RNA is similar to its weird cousin who lurks in the corner at family gatherings, DNA. It also has a sugar-phosphate backbone with nucleotide bases attached to it. There are 3 major differences between RNA and DNA. First, RNA is a single-stranded molecule. No, incorrect. There is no double helix. Yes, correct. Secondly, the sugar in RNA is ribose, which has one more oxygen atom than deoxyribose. Lastly, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. Now that we know all the basics of a Gummi bear's DNA structure, we need to understand how it copies itself, because our cells are constantly dividing, and that requires a complete copy of all that DNA information. That is called replication. cell in a gummy bear's body has a copy of the same DNA. It started from an original copy, and it will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. Helicase is very great, unwinding the double helix at speeds so fast even Miss Frizzle couldn't reach. It slices open those loosey-goosey hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication spoon. No, fork. Yeah, fork. That sounds better than spoon. It has a top strand called the top strand. Ha! Fitting. And another bottom strand called the lagging strand. Kinda like my ex-boyfriend. These unwound sections can now be used as templates to create two complementary DNA strands. But remember, the two strands go in opposite directions in terms of their chemical structure, which means making a new DNA strand for the top or leading strand is going to be much easier than for the lagging strand. For the leading strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down to the molecule. But before it can do that, it needs a section of nucleotides that fill in the section that's just been unzipped. Starting at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer so that it can start building the new DNA chain. And for that primer, we can thank the enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning. Then DNA polymerase just follows the unzipping, adding new nucleotides to the new chain continuously all the way down the molecule. Much more difficult. This is because DNA polymerase can only copy strands in the 5' to 3' direction, and the lagging strand is 3' to 5'. DNA polymerase can only add new nucleotides to the free 3' end of a primer. Since the lagging strand runs in the opposite direction, it has to be copied as a series of segments. Then the RNA primase lays down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, and each starting with an RNA primer called Okazaki fragments. These allow the strands to be synthesized in short bursts. Then another kind of DNA polymerase has to go back over and replace all those RNA primers. And then all the little fragments get joined up by a final enzyme called DNA ligase. DNA gets it wrong about 1 in every 10 billion nucleotides. But it turns out DNA polymerases can also proofread, in a sense, removing nucleotides from the end of a strand when they discover a mismatched base. And that is how a gummy bear's DNA do. This has been True Facts about the gummy bear.